Hello, everyone. Welcome back to the ISCB-SC webinar series. Before we start with the today's talk, I just want to let the audience know that they can follow us on Twitter. Our handle is @ISCB-SC. Uh, by following us on Twitter, you'll stay tuned with our upcoming webinars as well as learn about the opportunity to be part of our next webinar series. Now, without further ado, I would like to welcome our today's speaker, Sebastian Ayala Ruano. Sebastian is a research assistant at Universidad San Francisco de Quito in Ecuador, and today he'll be talking about exploring the chemical space of antiparasitic peptides and discovery of new promising leads through a novel approach based on network science and similarity searching. So, welcome, Sebastian, and the floor is yours now.
Okay, so hello everyone. Thanks Ayush for the great introduction, and thanks to the ISCB Student Council for the opportunity to share my research here. So I will start. So first, this is the presentation outline, which is divided into an introduction to the research topic, methods we applied, the results we obtained, and conclusions. So to start, we should know that in the last decades, antimicrobials have contributed to preventing and treating infectious diseases caused by bacteria, viruses, fungi, and parasites. However, inappropriate use of these medicines, poor sanitary conditions, and other causes have created an ideal scenario for the emergence of multi-drug resistant pathogens that are not treatable with the available drugs. In this cartoon representation, we could observe a summary of how this process works. So in this context, it's mandatory to search for new antimicrobials less susceptible to evolutionary resistance mechanisms. So antimicrobial peptides have appeared as good alternatives to control infectious diseases that have a slower emergence of antimicrobial resistance. AMPs are small bioactive compounds, commonly with fewer than 50 amino acids, amphipathic properties, and a net positive charge. The effectiveness of AMPs resides on the wide range of mechanisms they can use for both killing microbes and modulating immune responses. So AMPs could be used to treat infections caused by different microorganisms. In this project, we focused on the antiparasitic activity, which could help to treat malaria and neglected tropical diseases such as leishmaniasis, Chagas disease, among others. So the AMS chemical space is huge. It's estimated that there are more than 10 to the 65 unique sequences of peptides with 50 residues or fewer, which represent a big challenge for the discovery of new promising bioactive peptides. In this scenario, computational aided pipelines have been proposed as efficient alternatives for the high throughput screening of AMPs. Traditionally, strategies applied for the discovery of AMPs have relied on bioinformatics methods, pattern matching approaches like regular expressions, evolutionary algorithms, molecular fingerprint comparisons, and QSAR models. More recently, machine learning algorithms have been extensively used to predict and discover new potential AMPs. Most of the machine learning methods to predict AMPs have focused on supervised strategies, requiring labeled datasets to train these models. These supervised algorithms have shown some issues regarding the size, quality, diversity, application domain, and representative of datasets required to train the models, which could produce inappropriate predictions and wrong results. So, considering the limitations and drawbacks of the available method to discover AMPs, in this research we presented a novel approach based on network science and multi-query similarity searching models to discover new potential antiparasitic peptides. So our workflow was divided into four stages. First, the network analysis part, next, the multi-query similarity searching models, then the APP's prediction, and the discovery of sequence motifs. The first stage started with the data collection. We obtained the APPs from the StarPep database. This database is embedded in the StarPep Toolbox, a software with a graphical user interface designed to perform network analysis of peptides. Then we use this software to create three types of networks: chemical space networks, half space proximal networks, and metadata networks. So the uh, chemical space networks and half space proximal networks apply different methods for their construction, but both are similarity or correlation networks, defined as a set of nodes and a set of edges. In these networks, nodes represent APPs characterized by multidimensional sequence-based molecular descriptors, and edges or links are pairwise similarity relationships between descriptors of the peptides. Thus, the nodes of the chemical space networks and half space proximal networks are connected because they are similar to each other. In addition, the metadata networks are multi-layer networks defined as a set of nodes and edges, same as in the previous networks, but the metadata networks also have a set of layers representing different edge types or labels, which in this case are the metadata and the APPs. So metadata comprises additional information from APPs, such as their origin, database function, target pathogen, among others. So the second stage included the design of multi-query similarity searching models, in which we use the most central APPs by different centrality measures as queries against the StarPep database. We selected the best models by evaluating their performance in five benchmarking datasets of APPs and non-APPs. Also, we compared the performance metrics of our best models with machine learning based approaches to predict APPs. So the third stage was the repurposing of new potential APPs by applying the best multi-query similarity searching models and some filters of toxicity and hemolytic activities. And the last stage was the discovery of sequence motifs shared by the potential antiparasitic leads using multiple sequence alignments, alignment-free methods, and the ProSite server. So regarding the results, before creating the definitive chemical space network and half space proximal network, we conducted some analysis to decide the proper similarity threshold for both networks. We constructed chemical space networks and half space proximal networks of APPs, changing the similarity threshold, obtaining 36 networks in total, 18 for each network type. So there is a lack of a predefined standard values for this cutoff because it depends on the input data, similarity relationships, among other aspects. Therefore, it's recommended to define this threshold case by case. So we studied what would be the best cutoff values for both networks, considering some network metrics. As we can see in this plot, here in the x-axis, here is the similarity threshold, and in the y-axis there are different network measures such as density, average clustering coefficient, modularity, number of communities, and singletons. So based on all metrics from the networks with different similarity cutoffs, we found that the best similarity threshold for both types of networks was 0.65. The chemical space network shown in blue here with this similarity threshold was the local maximum point of the average clustering coefficient, and it presented intermediate values of density, modularity, number of communities, and singletons, which was the ideal point. We created the chemical space network and half space proximal network by applying the 0.65 similarity threshold. Then we obtained the giant components of both networks, and we constructed null models with the same number of nodes and edges of the giant components. So here we can see the visualizations of the chemical space network and the half space proximal network and their null models with nodes colored by their community and sized by their weighted degree or strength, which is calculated by summing up the edge weights of the adjacent edges for each node. So we can see that the real networks had an apparent community structure because the nodes tend to form clusters, which is absent in the null models. So we calculated some network metrics to measure in a formal way the difference between chemical space network and half space proximal network, as well as between real and null models in terms of their community structures and other aspects. So the density and the average clustering coefficient of half space proximal network had lower values than chemical space network, while the opposite occurred with modularity, communities, singletons, diameter, and the average path length. So this behavior is related to the way each network model is constructed and the number of edges obtained by each process. So comparing the real networks with their random model counterparts, as we can see in this table, modularity, number of communities, and average clustering coefficient values of the real networks were greater than the random models. Hence, the real networks showed better community structure and network connectivity. So regarding the metadata networks, this showed valuable information about the APPs. In the origin network, we observed that most of the APPs came from synthetic constructs, which is the largest node in this network, and a few of them were derived from parasites and bacteria. In the function network, it's shown that the most prevalent functions were parasitic, antiparasitic, and antimicrobial. And the target pathogen network shows that the most predominant pathogens targets were some bacteria such as Escherichia coli and Staphylococcus aureus, although we also found parasites such as Plasmodium, Leishmania, and Trypanosoma. Finally, the database network showed that the APPs came from different sources of information. So the centrality is a key concept in network science because it provides an intuition on the importance of nodes in networks. Therefore, we calculated the four centrality measures available in the StarPep software, which are the weighted degree, betweenness centrality, harmonic centrality, and hub-bridge centrality, which are different ways to calculate the importance of nodes. Then we calculated these values for all nodes from both networks, and we obtained the set of the most central peptides by each metric, which we are used to retrieve new potential APPs by similarity searching, as I will explain in the next slide. So here is the summary of the multi-query similarity searching models. Um, First, we have query datasets composed of singletons, which are nodes that are not connected to other nodes, and the most central APPs by the four centrality measures for both networks and the consensus set of both networks. So we also have reference databases, which are validated sets of APPs and non-APPs. So we calculated similarity scores with the Smith-Waterman local alignment algorithm between the queries and the database molecules. Then we obtain single fused scores, which correspond to the largest of all similarity scores for each query. And finally, we sorted the scores, applied a similarity threshold, and obtained the predicted APPs and non-APPs. And the performance results of the multi-query similarity searching models showed that the combination of query sets obtained with different centrality measures and the consensus of both networks was always better than any query set derived from a single centrality measure and type of network. Also, adding the singletons to the query datasets enhanced the recovery of models. So after selecting the best multi-query similarity searching models, we compared their performance with the machine learning based methods Discover and Fun. And we observed that our models had higher values than the machine learning based models in all the performance measures. Some regarding the repurposing of new potential APPs, our starting search space was the entire StarPep database, which contains about 45,000 peptides. After applying a series of filters with the StarPep toolbox, the best multi-query similarity searching model, and some external servers to delete toxic and hemolytic peptides, and for validating the annotation of these APPs, we retrieved 95 leads that have never been associated with the antiparasitic activity. So the figure on the left summarizes the filtering process applied to retrieve the set of new potential APPs, and the figure on the right depicts the process to reduce most of the peptides from the initial search space, applying our best multi-query similarity searching model. So the figure on the left shows the chemical space network of the 95 potential APPs with their communities, which exhibit the diversity that these peptides still have. So to measure such similarity among the repurposed leads, we calculated a pairwise sequence identity among all of these peptides, and we found that most of the sequences share pairwise identity values below 30%, as we can see in the heat map on the right, where most of the points are blue, which represent their low sequence identity. And to perform a wide exploration of motifs, the resulting 95 leads peptides were clustered, obtaining 5 clusters. Then we screened each of these clusters for discovering sequence motifs. And in this table, you can see the obtained motifs applying various multi-sequence alignment algorithms. Also, we identified motifs using other methods such as stream and prosite, but these results are not shown here. Regarding the conclusions, in this study, we created a novel approach based on network science and similarity searching to explore the APP's chemical space. And this pipeline outperformed the state-of-the-art machine learning models for APP prediction. And this study could support research aimed at repurposing peptides to unknown bioactivities. And here we proposed 95 repurposed potential APPs that have not been associated with this activity. And we found promising common motifs that can serve as templates for searching novel APPs. So I want to thank all the collaborators and research groups that contributed to this project, especially to Giovanni Marrero Ponce and Noel Perez, who gave me the opportunity to collaborate on this project and provided proper guidance throughout all the stages of the project. Also, thanks to the ISCB Student Council for creating spaces to share our work in bioinformatics. And finally, if you want to know more about this project, you can freely access the slides, the preprint of this project, and the StarPep database and software with these links. And if you have any questions, please let me know all of your questions now, or you can reach out to me by email or social media. So thank you everyone for your attention.
Thank you, Sebastian. That was such a great talk. Uh, really enjoyed it. Thanks. To the audience, I would like to ask you that you can post your questions in the comments, and Sebastian is here to answer them. In the meantime, I can start with maybe one question. So I'm not an expert in this field, so maybe it's a very basic question, but why do you think that your current model outperformed state-of-the-art machine learning methods? Because that was quite surprising for me. I thought machine learning is everywhere.
Yes, yes. For us, it was also an interesting result, and we think that this result could be because of the machine learning approaches rely on supervised strategies that require labeled datasets. So in labeled dataset usually had a lack of diversity in the sequences, or there is not enough information to train the machine learning models to obtain good results. So we thought that for these limitations of the machine learning, of the supervised machine learning methods, our method maybe was better than that. But if this is a temporary result, because maybe if in next, if there are more data sets to train machine learning models, maybe they can improve their results. But this
Okay,
is a new strategy that we proposed.
quite interesting. Thank you. Okay, so just a follow-up question on that. Do you think that your novel approach can in some way be combined with the machine learning methods to improve predictions?
Yes, I think that we could integrate both the strategies to maybe provide better predictions and discover antiparasitic peptides, or in general antimicrobial peptides more accurately. So yes, I think that it could be better to combine these approaches. And it's interesting because in previous works in this field, um, some researchers have combined machine learning strategies with other methods, such as bioinformatics approaches based on sequence alignments, or also with evolutionary algorithms or QSAR models. So yes, I think that in the future we could think to merge these strategies to obtain better results.
Thank you. So, uh, I guess we still don't have any questions from the audience. But I would like to mention that everybody in the audience can reach out to Sebastian. He shared his social media accounts, so you can always talk to him about the work. So finally, thank you. Thank you very much, Sebastian. And we hope to see you again in the next season of our webinar series.
Oh, thanks everyone, and thanks Ayush for the great hosting.
Thank you.